My name is Miquel Pons. I am a professor in the University of Barcelona. I am the scientific director of the NMR facility and a group leader in biomolecular NMR. We are interested in proteins, mostly. Basically, we work in intrinsically disordered proteins. Intrinsically disordered proteins are proteins that do not have a fixed shape, but they are moving, and therefore they are ideal for NMR. And those proteins have to be involved in cancer. And also, we are also interested in other kinds of proteins of, let's say, biomedical interest, like biologics of different kinds, so large molecules that are used as drugs. The things for which a GHz instrument is useful, especially, is whatever thing needs a very high resolution. Of course, ITPs, so the intrinsically disordered proteins of several types, they are important. Also, very high complexity molecules. So, for example, when you talk about an antibody, it has a very large number of signals, and that leads to overlap. And of course, for this kind of complex samples, you need to have the resolution that you can achieve with a GHz. I mean, NMR is the most versatile technique, I would say. When I speak to non-specialists, what I explain is that with NMR, we talk to the atoms. Okay, so whatever complex is your system, I mean, you can really get information directly for each of the individual atoms that forms your complex systems. And of course, no other techniques can do that. Basically, more resolution. I mean, we can tackle more complex problems that we could use in the past. I mean, we've gone from studying part of a molecule, so we had to cut our problem into smaller problems, but now we can study the whole protein in the GHz, and that it's very important because, of course, I mean, divide and conquer, it's good, but it has limitations. At some point, you do have an interplay between different parts of the molecule, so that you can only get this information when you talk with the whole protein, and for that you need higher field instruments. Actually, having a high field instrument allows you to study more complex molecules. Why do you want to study more complex molecules? Well, because at some point, the function of the molecule may depend on the interplay between long-distance regions. For example, we have just found out that the intrinsically disordered region of one protein we are studying, in fact, is folding back all the way to the other part of the protein. Until now, we were studying it independently. We knew it was doing things, but we didn't know that what it was doing was in fact reaching out to the far end of the protein. And for that, we only got this information because now we are able to study the entire protein, and the complexity of this protein, it's a 60 kilodalton protein, would prevent us from studying at a lower field. And so this has made, that's an example that made a big difference for us. Nowadays, our main concern is about intrinsically disordered proteins. Some years ago, people didn't know that they exist because, for example, by X-ray, those are invisible. So people thought that they were not important because it was believed that you had to fold the proteins to have function, and therefore something that was not folded would not be functional. Nowadays, we know that maybe 2/3 of the proteins, of human proteins, have disordered regions. This is something that happens to humans, not to bacteria. So humans are special. So our complexity derives from the fact that we have disordered proteins. And disordered proteins can only be studied at atomic detail by NMR. So if you want to study human proteins, you have to use NMR. I mean, if you don't care about human proteins, if you just care about bacterial proteins, you may forget about NMR, or you may not need it. But if you say that your interest is in human proteins and therefore in human diseases, then NMR, you cannot avoid it. It's your technique. we found, this is still unpublished results, so I'm just explaining the general thing, but one special modification of the disorder region that affects the way in which cancer cell divide, that occurred was known, but why it occurred was not known. Now, by NMR, we have now demonstrated how this very important thing happens, and that of course opens the way in which you can try to design drugs to correct problems arising from this kind of interaction. So it's the divide between basic research and applied research is very thin, and especially when you talk with NMR, because NMR senses the functional things in proteins. So it's not just that you play with NMR to just have a nice picture of the protein. You want to know how it works. And therefore, if you know how it works, for a human protein involved in a disease, that immediately means you may use this information for translation, but we certainly can focus our attention in places where the problems are, and therefore we have the possibility to help in solving those problems. My vision is that we eventually will be able to understand how disordered proteins work. I mean, for the moment now we are happy to know that they are important. They are complex, they are very complicated. I mean, we are not at the stage where after 50 years, we know how to design, predict what a folded protein will do. For disordered proteins, we still do not have that. Now, I think this is the goal. So we have the next conceptual step is that we are going to understand, and we are going to be able to predict, and we are going to drug disordered proteins. And in order to do that, NMR is going to be important, and because disordered proteins tend to be crowded, because there is very little dispersion in the spectra, working at high fields is essential. So for in the field of disordered proteins, high field NMR is very, very important. In order to advance, you have to take risks. I mean, the particular story of our GHz is that we already had commissioned the instrument. We had to wait for several years until it was delivered, and then we were offered the option to say, okay, would you like to take the risk of testing this new kind of magnets with high temperature per 1 GHz? Nobody has ever tried that. So the first one in Europe, the second in the world, came to Barcelona. We are extremely happy with that. So it means we took a risk, but I think that's the way you advance.